When we are analyzing our basidiocarp, remember that we want to analyze the underneath of the pileus. So we want to analyze the underneath of the cap. The underneath of the cap is referred to as the hymenium. Remember, the hymenium is the most superficial layer. It contains the spore-bearing structures known as the basidia. Here we can see the sterigmata or the sterigma, and we can see these potential basidiospores, which are going to be released into the environment. Right below the hymenium, we have the subhymenium, which is going to provide that structure and support for the hymenium to grow. And underneath the subhymenium, we have the trama, which is going to be the fleshy structure of our fungi. So the trama can actually have 4 different variations, and the trama can actually be viewed under a microscope to understand which kind of trama we have, what kind of fungi we're looking at, and etc. So first we have divergent fungi. So divergent fungi is going to be very, very similar to convergent fungi. They're essentially just moving in different directions. I like to think about it as divergent is going down, whereas convergent is going upwards. Now these two are quite similar. And then we have parallel trama. Parallel trama is literally going to be these parallel blocks, and they're all parallel to one another. And then lastly, we have interwoven trama. Now, interwoven trama, super straightforward. It just means that they are all interwoven with one another. So those are the 4 different kinds of trama that are found within basidiocarps. Remember that this trama can be analyzed underneath a microscope, and it is the most deep layer when we're considering the layers of the basidiocarp.